Good afternoon, everyone. I'm Sapna Tripathi, Assistant Professor, IIMT College of Pharmacy. Today, we will discuss about the topic immunity. So, what is immunity? Immunity is the ability of the body to defend itself against disease-causing organism. Every day, our body comes in contact with several pathogens, but only a few results into diseases. The reason is our body has the ability to release antibodies against these pathogens and protect the body against diseases. This defense mechanism is called immunity. Now, there are mainly two types of immunity. The first one is innate immunity, and the second one is acquired or adaptive immunity. Innate immunity is present in an organism by birth. This is activated immediately when the pathogen attack. Innate immunity includes certain barrier and defense mechanism that keep foreign particle out of the body. Innate immunity refers to the body's defense system. This immunity helps us by providing the natural resistance components, including salivary enzymes, natural killer cells, intact skin, and neutrophils, which produce an initial response against the infection at birth prior to exposure to a pathogen or antigen. It is a long-term immunity in which our body produces the antibodies on its own. Our body has a few natural barriers to prevent the entry of pathogen. The innate immunity are mainly of three types: species immunity, racial immunity, and individual immunity. Species immunity shown by all members of a species against pathogen. For example, birds are immune to tetanus. Next one is racial immunity. Races show marked difference in their resistance to certain infectious diseases. And the third one is individual immunity. This is very specific for each and every individual, despite having same racial background and opportunity for exposure. The second type of immunity is acquired immunity, which is also known as adaptive immunity. Adaptive immunity is the immunity that our body require or gain over time. Unlike the innate immunity, this is not present by birth. The ability of the immune system to adapt itself to disease and to generate pathogens specific immunity is termed as acquired immunity. It is also known as adaptive immunity. It is specific and mediated by antibodies or lymphocytes which make the antigen harmless. The main function of acquired immunity is to relieve the victim of the infectious diseases and also prevent its attack in future. Now, the acquired immunity is also of two types: active immunity and passive immunity. Active immunity is induced by natural exposure to a pathogen or by vaccination, and the passive immunity is achieved by transfer of immune products such as antibody or sensitized T cell from an immune individual to non-immune one. This is the flow chart for immunity. You can easily classify immunity into two parts. There are two other types of immunity: humoral immunity, and the second one is cell-mediated or cellular immunity. Humoral immunity is defined as the immunity mediated by antibodies which are secreted by B lymphocytes. B lymphocytes secrete the antibodies into the blood and lymph. The blood and lymph are the body fluid. Since the B lymphocyte provide immunity through humor, this type of immunity is called humoral immunity or B cell immunity. Antibodies are the gamma globulin produced by B lymphocytes. These antibodies fight against the invading organism. The humoral immunity is the major defense mechanism against the bacterial infection. Humoral immunity, also known as antibody-mediated immunity. It involves production of antibodies against foreign antigen. Antibodies are produced by a subset of lymphocytes called B cell. And the second type of immunity is cell-mediated immunity. It involves specialized set of lymphocytes called T cells and recognize foreign antigen on the surface of cell organism. There are two type of cells, helper T cell and cytotoxic T cell. T cell regulate proliferation and activity of other cell of the immune system, B cell, macrophages, neutrophils, etc. and defense against bacteria and viruses that are inside host cell and are also in protozoa, also in cancer cell and transplanted tissues. Now here come the next topic, which is immunoglobulin. As you know, immunoglobulin is also known as antibodies. Immunoglobulin is defined as it is a glycoprotein which is produced by plasma cells, or you can also say any class of protein present in the serum and cell of immune system which function as antibodies known as immunoglobulin. And the third definition is the protein that is made by B cell and plasma cell and help the body fight against infection is known as antibodies or immunoglobulin. This is the structure of immunoglobulin. This is a Y-shaped structure of immunoglobulin. Uh, if you talk about the structure of immunoglobulin, it is composed of 4 polypeptide chains, which consist 2 identical light chains and 2 identical heavy chains. These chains are linked by disulfide bonds and by non-covalent interaction. Light chain similar in all immunoglobulin. Light chain occur in two varieties, two type of constant region sequence, kappa and the lambda. In a particular antibody, either 2 lambda or 2 kappa chains are present, but not 1 lambda and 1 kappa. Light and heavy chains are subdivided into variable and constant region. Each heavy and light chain contain amino terminals in variable region and carboxy terminal in constant region. Next, if we talk about the classification of immunoglobulin, there are mainly 5 primary classes of immunoglobulin: IgG, IgM, IgA, IgD, and IgE. It is classified according to their chain structure, if it is light chain or heavy chain. The first one is IgG. It is in monomer structure. If you talk about the percentage serum antibody, it is 80%. The location of IgG immunoglobulin is blood, lymph, and intestine, and the half-life is 23 days. Next one is IgM. Structure of IgM is pentamer, and the percentage serum antibody is 5 to 10%, and the location of IgM immunoglobulin is in blood, lymph, and B cell surface, and the half-life is 5 days. Next one is IgA. It is dimer in structure. Percentage serum antibodies is 10 to 15%, and the location is tears, saliva, intestine, milk, blood, and lymph, and the half-life is 6 days. The next one is IgD. The structure of IgD immunoglobulin is monomer. The percentage serum antibody is 0.2%, and the location of IgD immunoglobulin is B cell surface, blood, and lymph, and the half-life is 3 days. And the last one is IgE immunoglobulin. The structure of IgE immunoglobulin is also monomer, and the percentage serum antibodies is 0.002%, and the location of this immunoglobulin is It is bound to mast cell and basophil throughout the body and in blood. And the half-life in serum is 2 days. Next, here comes the next topic, immunostimulants. Immunostimulant are also known as immunostimulator. These are the substances that stimulate the immune system by inducing activation or increasing activity of any of its component. Immune stimulation refers to the stimulation of immune system by an external source. This stimulation can confer a protective effect against microorganism. As well, immune stimulation shows promise as a means of obtaining an immune response to conditions such as cancer. Conventionally, the immune system is stimulated into producing antibodies or other infection fighting constituent in response to an infection. There are two main categories of immunostimulants. First one is specific immunostimulant and the second one is non-specific stimulants. Specific immunostimulants provide antigenic specificity in immune response such as vaccine or any antigen, and the non-specific immunostimulant act irrespective of antigenic specificity to augment immune response of other antigen or stimulate component of the immune system without antigenic specificity, such as adjuvant and non-specific immunostimulators. Next one is immune suppression. Suppression of the body's immune system and its ability to fight infection and other diseases. Immunosuppression may be deliberately induced into drugs as in preparation for bone marrow or other organ transplantation to prevent rejection of the donor tissues. It may also result from certain diseases such as AIDS or lymphoma or from anti-cancer drugs. What are the causes of immunosuppression? Immunosuppression may be caused by a number of systemic illness. These include, but are not limited to these parameters. Diabetic mellitus, renal or hepatic failure, infection of the central nervous system, systemic lupus erythematosus. Next one is rheumatoid arthritis. Now, what are the causes of immunosuppression? Immunosuppression may be caused by a number of systemic illness. These include these parameters which we already talked about. In addition to systemic illness, certain pharmaceuticals and therapeutic intervention can also cause immunosuppression. For example, corticosteroids, immunoglobulin, antimetabolites, biological alkylating agents, and ionizing radiation. These all are the these all are the causes for immunosuppression. Thank you. That's all for today's lecture.